Hello, dear students. So, we'll discuss today about the O-link glycosylation. We discussed about the N-link glycosylation before. O-link glycosylation, this is a type of glycosylation which will happen wherein the sugars are going to be added to serine or threonine hydroxyl groups. This particular O-link glycosylation is little different from that of N-link glycosylation, not just in terms of the glycosidic bond which get formed, but also in terms of addition of sugar units, because N-link glycosylation is going to happen wherein a pre constructed oligosaccharide unit will be added onto the protein, and followed by that there will be trimming and as well as processing of the oligosaccharide unit. But here in case of Wolin glycosylation, it is not going to happen in that particular way, where here the addition of oligosaccharide units, addition of the saccharide units or the sugar units will happen one by one, and it involves the glycosyl transferases which are going to work in a stepwise manner, and they are going to be highly specific, not just only in the addition of particular type of glycosyl moiety, but also in the type of specific sugars, as well as the substrates which they are going to use for the transfer. And also they are very specific in terms of what type of the glycosidic bonds they are going to get involved in the formation. Either they form in the alpha 1,4 or alpha 1,6 or beta 1,4 or beta 1,6 or beta 1,2 or alpha 1,2, like that. Very specific with respect to even the carbon atom wherein the bonds are going to get established. So these enzymes also they are involved or located in various compartments of Golgi operators, and each glycosylation involves appropriate nucleotide sugars. So this particular glycosylation is going to get initiated and completed within the Golgi apparatus. You can see this is the one type of glycosidic bond. There upper you can see it is the beta galactosyl 1,3 N acetyl glucosamine serine threonine, that is one type. Another way is M manosyl, alpha manosyl serine threonine, that is another type of glycosidic bond, which will happen with the help of sorry with the help of two different enzymes. So if we come to the mechanism of addition of sugar units, so here I am giving you a mechanism of addition of sugar units wherein the proteins are going to be first trafficked into the Golgi apparatus. That means they are going to get vesicularized from the endoplasmic reticulum, and then they will be transferred into the cisternae of the Golgi. And in the Golgi complex, okay, initially in O-link glycosylation generally happens with the first addition of N-acetyl galactosamine, wherein the N-acetyl galactosamine is added by the N-acetyl galactosamine transferase enzyme. This transfers a single N-acetyl galactosamine unit to the beta hydroxy of the serine or threonine. Some proteins are glycosylated with N-acetyl glucosamine, or sometimes even fucose, galactose, mannose, xylose are also. The sugar nucleotides which are going to be used here, they are generally going to be the monosaccharide donors which are going to be added here. So here in this particular picture you can see how these particular sugar units are being added, okay, to the polypeptide chain. So for example, first the N-acetyl galactosamine is being added, followed by that N-acetyl glucosamine in a stepwise manner. There will be UDP, N-acetyl glucosamine, then followed by UDP galactose, then there will be CMP sialic acid, like that. One by one these particular sugar units are going to be added into the polypeptide chain, establishing a specific type of glycosidic bond and constructing a sugar tree on it. So that's how the O-linked glycosylation is going to takes place. The next type of glycosylation is glycation. So this glycation is nothing but this is the formation of GPI, that is the earlier which I spoke about, the glycosyl phosphatidylinositol. In this glycation there will be linking of the sugar, as well as the sugar is going to act as a linking between the protein and the what we call lipids. So in GPI anchoring, it consisting of phosphatidylethanol-amine linker that binds to the C terminus of the target protein. So here it also involves a glycan core and then phospholipid tail that anchors the structure to the membrane. So this is the GPI structure you can see, in which the glycosyl moieties, the glycan core is linking the protein through the phosphatidylethanol-amine to the phospholipid tail, which actually anchors itself into the membrane. And some of the GPI linked proteins are going to be acetylcholinesterase, alkaline phosphatase, and there is decay accelerating factor, and even 5' nucleotidases are there, and thymine Th1 antigen is also an example for GPI linked protein. So here the after glycosylation there is going to be other types of modifications which we are going to observe in the proteins, that is sulfation, where addition of sulfohydryl group is going to happen, or sulfate molecule is going to take place for mannose and N-acetyl glucosamine residues, so that they can convert into glycosaminoglycans, which are components of the proteoglycans, that are sulfated heteropolysaccharides, which we have already seen earlier. Then followed by that there is acetylation of sialic acids. So this is facilitates the protein-protein interaction. The acylated sialic acids are going to help in that. Then there is phosphorylation, in such as the mannose residues are going to get phosphorylated to form mannose 6 phosphate, which ensures the trafficking of some of the specific proteins into the lysosome. That is, lysosomal proteins are going to get trafficked even based on the mannose 6 phosphate residues. So these are a few of the modifications which are going to happen. Further protein modifications are also involves the formation of disulfide linkages, that is proper disulfide linkages, because disulfide linkages, if they are not going to get formed properly between the subunits or within the particular polypeptide chain, it is not going to facilitate proper folding of the proteins. It can leads to improper folding or misfolding of the proteins. So therefore protein disulfide isomerase enzyme is going to play an important role. It generally forms disulfide bond within the endoplasmic reticular lumen, wherein it's going to arrange. First there will be reduction of the oxidation of the sulfohydryl group of cysteine amino acids, which involves in the formation of disulfide linkage, and later this particular disulfide isomerase, it isomerizes, means it alters this particular disulfide linkages and establishes the proper disulfide linkage and rearranges it. So thereby it forms specific required type of disulfide linkage, so which results in the proper folding of the protein and results in the formation of a functional protein. So this particular pathway of formation of disulfide linkage in case of eukaryotes, it happens within the endoplasmic reticulum. You can see it is also facilitated with by a protein called as Euro1, which actually helps in the formation of the sulfhydryl groups by within the PDI, which can further form the proper disulfide bond. And in case of bacteria, the same function is going to be done by the DSB1, DSBA, which is going to act as PDI, and DSBAB, which acts as ERO1 protein molecule in case of the bacteria. And in case of bacteria, the process will takes place within the periplasmic space. Okay, in case of eukaryote, it happens in the endoplasmic reticular lumen. Then once the folding of the particular protein is done, then there is going to be requirement is, in case if the protein is a multimeric protein, it need to undergo multi subunit arrangement. So this arrangement of multimer or assembly of the multimer is going to take place. It depends. Sometimes it can happen within the endoplasmic reticulum. Sometimes even it can happen during the in the Golgi complex also. So this arrangement in case of endoplasmic reticulum specifically happens for the membrane protein. For example, here it is shown the hemagglutinin protein, which is a trimeric protein molecule having three finger-like projections. So this particular, each of the subunit is a membrane protein. First it forms this synthesis of membrane protein takes place, and then initial glycosylation happens within the endoplasmic reticular lumen, and followed by that, then it's get inserted also into the membrane, then followed by that this particular protein subunits, there will be formation of three of these subunits, and then later these three of them come together through the help of chaperonins and other proteins, and they form a trimeric assembly. Whenever it is in case of hemagglutinin protein, it is going to lead to formation of trimeric assembly, so thereby multimeric subunits are going to get formed. Similarly, in case of immunoglobulins, wherein it has two heavy chains and two light chains, so their assembly is going to happen such a way that first there will be formation of the heavy chains, then there will be light chains. The heavy chain first combines with the light chain and form heavy chain light chain dimeric assemblies, and these heavy chain light chain dimeric assemblies then they assemble with each other to form heavy chain light chain heavy chain light chain tetrameric assembly, which is going to result in the formation of a functional immunoglobulin. If the immunoglobulin is a membrane protein, so then in the same similar fashion that immunoglobulin membrane protein is also going to get assembled. So what in case if the protein is a misfolded protein? So in improperly folded protein, if it is going to get formed, then this improperly folded protein induces the formation of chaperonins, and these chaperonins further they help in the proper folding of the protein molecule. So the first the protein gets unfolded, and incomplete folded proteins they get into the endoplasmic reticulum. So once they get into the endoplasmic reticulum, the chaperonins like BIP is going to get released, and this release of BIP is going to happen from a protein called as IR1, that is IRE1 protein. And once BIP get released, this BIP is going to unfold the proteins, means it's going to completely linearize that particular proteins, acts as a chaperonin, and also activates this particular IRE1 protein. In turn also involves in the activation of translation of other kind of chaperonins like HSC1 mRNA, or it can be other HSPs are going to get synthesized. These chaperonins then again get transported into the endoplasmic reticular lumen, wherein they further fold the protein molecule into a proper three-dimensional structure. In case if the misfolded or unfolded protein molecules, if they are not going to get properly folded, then they'll get transported back into endoplasmic reticulum, wherein the protein degrading machinery will take that particular polypeptide chain through either ubiquitination or by recognizing the misfolded protein recognizing proteins. They get bound to it and they carry to the proteosomes, where the degradation is going to get happen. So last type of protein modification is protein cleavage. So this cleavage is also going to be an essential process, wherein a pre-pro protein can get converted into pro protein and then protein. So which we generally see it in case of insulin and other hormonal protein molecules. So constitutively secreted protein molecule, so that is, for example, in case of albumin, it synthesizes as a proalbumin. So this proalbumin will have a small polypeptide portion, which is actually a protein. That means it is not required for the activity of the protein, but it just stabilizes the nascent polypeptide chain. So this protein part is going to get cleaved out by the furin endoproteases, which removes the protein part, leaving only the albumin molecule, so which can again get folded and get transported into the means get secreted out of the cell. In case of insulin, it gets synthesized as a proinsulin, become which consisting of both heavy and light chains and C peptide together. Then by the enzyme endoproteases, so the C peptide will get removed and leaving the A chain and B chain separately. A chain and B chain further, they are going to involve the formation of insulin with the removal of the the arginine amino acids from the A chain, sorry, from the B chain by carboxypeptidases. So like that, proinsulin gets converted to the insulin by proteolytic cleavage. So these are some of the important transportations and signal sequences which will happen in the protein synthesis. Like endoplasmic reticulum, it will have N-terminal signal sequence. Okay, so the location sequence within the protein is N-terminal, so it consisting of about 6 to 12 hydrophobic amino acids, which specifies the movement of protein into endoplasmic reticular lumen. Like similarly, mitochondria, chloroplast, proteosomes, they all having a particular sequence of amino acids at their N and G terminals, which specifies the movement of proteins into specific destinations. So I'll stop here. I'll continue later. So with the export of proteins, how it will happen into the in case of bacteria.